Hello boys, welcome back to another session of video tutorials, and this session is meant for class 11th biology students, and this is meant for 11th August 2020. If you recollect from your memory, you are able to understand that we were talking about Kelvin cycle, and in the Kelvin cycle, as all of you know, there are three steps. Step number one is carboxylation, and in carboxylation, CO2 combines with RUBP, ribulose 1,5-biphosphate, with the help of an enzyme Rubisco. It results in the formation of two molecules of PGA. You can see here 3-phosphoglycerate, or it is also called 3-phosphoglyceric acid. This is the first stable product of carboxylation. And second, we were talking about reduction. The process involves in the step or reversal of the process found in the steps during glycolysis of respiration. Here the PGA is reduced to produce glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate, a key product used in the synthesis of both carbohydrates and fats. Then third step is regeneration. As you are seeing, in the regeneration, it involves a series of reactions that results in the formation of carbon acceptor RUBP and make it ready to accept another molecule of CO2. So the net reaction of C3, dark fixation of carbon dioxide, is 6 molecules of RUBP plus 6 molecules of CO2 plus 18 ATP plus 12 NADPH will give back 6 RUBP, because regeneration of RUBP is a compulsory, and C6H12O6, that is glucose and starch that we are writing here. The main aim of the photosynthesis is starch production, and then 18 ATP is converted to 18 ADP into inorganic phosphate, and 12 NADPH becomes 12 NADP plus. So this is the overall net reaction of C3 dark reaction or dark phase. Now you must also remember that whenever in Kelvin cycle, of course in your textbook it is there, you remember that two molecules of 3-PGA, 3-phosphoglyceric acid, as I have written here, one side 3-PGA, another side PGA. This 1-PGA will become 3-hydroxyacetone phosphate, 3-hydroxyacetone phosphate, and this gets converted to fructose 1,6-biphosphate. You can refer in your textbook, all these are there, fructose 1,6-biphosphate, and this fructose 1,6-biphosphate will become fructose 6-phosphate. Fructose 6-phosphate becomes glucose 6-phosphate, and glucose 6-phosphate will become sucrose and starch that you are seeing. So these are all changes that take place in Kelvin cycle, or it is also called a C3 cycle. And we said that there are plants in which instead of PGA there will be oxaloacetic acid, OAA we said. It's a 4-carbon one. So where the plants are having the C4, carbon 4 number, we call it as C4 pathway, and the plants are called C4 plants. These are also, this pathway, C4 pathway, is discovered by two great scientists, Hatch and Slack, HEATCH Hatch and SLACK Slack, Hatch-Slack pathway. So first time it is the Kochak and others reported that rapidly photosynthesizing sugarcane leaves produce a 4C compound like aspartic acid or malic acid as a result of carbon dioxide fixation. This was later confirmed by Hatch and Slack in 1966, and they reported that a 4C compound, that's why we are using that, 4-carbon compound called OAA is the first stable product. What about in C3? It is PGA, phosphoglyceric acid. Here OAA, oxaloacetic acid, is the first stable product in carbon dioxide reduction process in tropical grasses. So this led to an alternative pathway of carbon dioxide fixation, which is known as Hatch-Slack pathway or C4 pathway. And this is also called C4 cycle, as 4C compound is first stable product, and Hatch-Slack pathway, HSK pathway. This pathway was first reported in tropical grasses like members of family Graminae like sugarcane, maize, sorghum. In all this, C4 pathway will take place. But later in other subtropical plants like Altriplex, Amaranthus also has shown C4 pathway. C4 plants have one special anatomy, it's called Cranch anatomy, in which vascular bundles are surrounded by large sized bundle sheath cells. See, the size of the bundle sheath cells are very large, which are arranged in wreath-like manner in one to several layers. The chloroplasts of the mesophyll cells lack starch, but they have this. Mesophyll cells are specialized to perform light reaction, evolve oxygen, and produce assimilatory power ATP and NADPH. They also possess an enzyme called pepcase. There it is ribisco for initial fixation of carbon dioxide. The chloroplasts of bundle sheath cells are agranal. There is no grana in that. That's the case. Therefore, ATP is synthesized through cyclic photophosphorylation only, and photolysis and oxygen evolution is absent. So bundle sheath cells possess ribisco and are well protected from oxygen being released from mesophyll cells. And the primary acceptor of CO2 is PEP, phosphoenol pyruvate. You can see Cranch anatomy, Hatch-Slack pathway. You can read all this, and this is what I am telling. The process starts in mesophyll when CO2 is accepted by primary CO2 acceptor, which is 3-carbon molecule PEP, phosphoenol pyruvate. There it is RUBP. This is catalyzed by PEP carboxylase or pepcase. So mesophills lack ribisco enzyme. They don't have that. So C4 acid OAA is formed in the mesophyll cells, which then forms other 4-carbon compounds like malic acid, aspartic acid in the cells. Malic acid formed are transported to the bundle sheath cells. So once they are transported to bundle sheath cells, what happens? The malic acid will become a 6-carbon intermediate. Then it becomes phosphoglyceric acid, which is 2 molecules. The phosphoglyceric acid, you know it. The PGA enter into Kelvin cycle, and the Kelvin cycle results in regeneration of ribulose biphosphate. So this is a cycle. So cyclic thing keeps on happening. So in bundle sheath cells, these are broken down, as I said, to release CO2, and the 3-carbon molecule is transported back to mesophyll, where it is converted to PEP, thus completing the cycle. The CO2 released in the bundle sheath cells enters the C3 or Kelvin pathway. So Kelvin pathway is integrated one in this Hatch-Slack pathway, a pathway common to all plants. The bundle sheath cells are rich in an enzyme, ribisco, ribulose, biphosphate, carboxylase, oxidase, but lack pepcase. Thus, the basic pathway that results in the formation of the sugars, the Kelvin pathway, is common to both C3, C4. You can see here how this is taking place between bundle sheath cell and mesophyll cells. And what is common here? You can see fixation of Kelvin cycle. This is common. So you understand that the both for C3 and C4 plants, Kelvin cycle is a must. And you can see here atmospheric CO2 is getting into mesophyll cells, then phosphoenol pyruvate, then phosphoenol pyruvate getting fixed, C4 acid is formed, and this is transported through plasmodesmata from mesophyll to bundle sheath. From there again C4 decarboxylation, CO2 is removed, and this goes into Kelvin cycle, and this again phosphoenol pyruvate PEP is formed, and that gets transported and regeneration of ribisco, and it goes back from bundle sheath cells to mesophyll cells. So what you have to do, draw the diagram of this, keep your